Let me start today's lecture with a story called The Bull and Goat. One day there was a bull caught in sight of a lion who's chasing him vigorously, and he found a goat shelter and entered inside. You can predict both of the organisms have the horns. They started fighting with each other. Particularly goat doesn't allow the bull inside, and somehow bull is strong one and it has able to keep keep itself inside the shelter. And the bull finally fed up. It told that I'm not here to fight with you. I'm scared of the lion outside. Let me here for some time. Once the lion passes away, I'll show you what I am. The moral is one should never approach mean people when one is in distress. Hello Shishyas, welcome to lecture 6 on the cell division meiosis. The meiosis is one of the important cell division that only happens or occurs in the sexual cells of all organisms, mostly seen in the multicellular organisms, very rare or absent in the unicellular organisms. Now, let me give you a brief introduction about this meiotic division. The meiotic division is discovered by the scientists Oscar Hartwig and Benden. I think it is one of the oldest discovery. The Hartwig discovered in C. urkin X, and then Benden discovered recently, I think in 1900s, in Ascaris worm. So this way, these scientists contributed, but the term meiosis was given by Farmer and Moore. Interestingly, the term meiosis itself says that the division is to halve or divide. That's why it's called as meiosis or meiosis. Now we'll try to learn more about the importance. Meiosis is like division happens or occurs for twice. That's how we can simply refer it. First division is nothing but heterotypic division. So when you say heterotypic division, the division is going to decrease the number of chromosomes. If you have looked here, the 2n is going to form only single. That means when the division takes place twice, and it's going to reduce the number. So first division is going to reduce the number of chromosomes into 1, and that's why it is called as the heterotypic division. And next is called as homeotypic division. The homeotypic division almost resembles like mitosis. You do not see any changes in case of the chromosome. It just duplicates the chromosomes in way of the mitosis. And one more major difference is, if you can recall, the G0, G1, S and G2 phase. In meiosis, in the first step of meiosis or meiosis 1, you can see the synthesis phase, and you do not see synthesis phase happening twice. This is also one more reason why the chromosomes are not getting double number. That's why you can see the chromosomes numbers are coming down because of synthesis phase is absent completely. Let us roll. I found a beautiful it's quite difficult to find exact scientific presentation. The reason is it's quite limited, and only a few teachers can be able to do that in a well manner. Okay, we have to this is a presentation I found quite relevant. You can see here, mostly this is seen in case of producing gametes, and then the chromosomal number is going to get reduced. That means the contribution from parents are going to be this makes thing are going to be given to the children. So the father contribution will be single set of chromosome, and mother's chromosome is single set, and it's going to contribute to 2n. So this also makes sure that the offsprings are going to give the exact number of, or it's going to maintain the normal number of chromosomal sets. Alright, next we can look at the terms. The terms have already been introduced in the previous division, but somehow I'll give a quick revision over here. The chromatids are nothing but the strands. The strands you can see in the chromosome. So when you say this is a centromere, these are the chromatids. They're duplicated in synthesis phase, right? That's why they have two sets. So the chromatid is nothing but single strand which are joined by centromere. These are centromere region, and then sister chromatids means, so let's say these are set from the father, and when you come to the when you assume that there is another set, if you draw with another color, so another set from mother, so it can be represented like this. So this is from mother. You can see these chromosomes, these two are called as sister chromatids, and when you see here, uh, these two are compared as non-sister chromatids. That means the two different origins are considered as non-sister, the same origins are considered as the sister chromatids. Next is homologous chromosomes. The homologous chromosomes have specific length and centromere position, and also they possess similar genes, same characters corresponding to loci. Loci refers to the position where the chromosome lies. When you say the loci, let's say if you consider the characters, let's say the character of hair color, or you can see the similar kind of gene will be present here also. So this way the organisms are going to have the same set of characteristics on a specific gene. Alright, that's what the homologous chromosomes. Remember, the homologous chromosomes have similar origin, different origin, but similar shape, structure, but the source of the chromosome is different. Right, let's look at the next one. The crossing over. So the crossing over is major step. We believe that the meiosis causes a lot of blending features. That's the reason due to the crossing over. In crossing over, the genetic material will flow from one chromosome to another chromosome. So that is the reason why the chromosomes have the features of both. It will have half of the parents' features and half of the mother, father, and even could be sometimes ancestral as well. That's what mainly results in the crossing over. And next is chiasma or chiasmata. You can see the X-shaped linkages. So these X-shaped linkages are nothing but chiasmata. So this is the region where exactly the crossing over or the material exchange, the genetic material exchange is going to take place. That's how chiasmata is defined. And next, is the overview. We'll go to the meiosis one. I told you the heterotypic division is also called as meiosis one, the first set of divisions in the meiosis. So it is going to be suffixed as meiosis one. You can see this division is where called as reductional division. Here the number of chromosomes is going to come down. So it has 4 different substages: the prophase one, metaphase one, anaphase one, and telophase one. If you want to learn easily, you have to understand the prophase one and then the anaphase one the metaphase one very closely. Once you understand the prophase, metaphase, and anaphase partially, you will be very good at meiosis, because all other steps resemble like meiosis except these three. And the prophase 1 alone is classified into 5 different stages, 5 different substages. So this is really a big step, I can say, and it takes quite long. The first is called as leptotene, zygotene, pachytene, diplotene, and diakinesis. Here you can see the chromosomes are going to reduce its number. In the beginning, what happens is the chromosomes are going to start thicken. The chromatids are going to thicken and they appear in this shape. Okay, that's why the chromosomes get thicker. And next you can see the 5 different substages of prophase 1. Remember the sequence of the stages as well. Sometimes they may ask you. The leptotene, it will have the nuclear envelope getting disintegrated gradually, and then you can see the chromosomes, the red, let's say one from mother and the one from father. These are going to start condensing. You can see the centromere appearance is just showing. The centromeres are gradually being formed. And then you can see the zygotine stage. The zygotine stage where the bivalents are forming. The bivalents means you can see the chromosomes of both. So one from mother, one from father are going to overlap with each other at particular points. Okay, this is where the synaptonomal complex is going to formed. The synapses where exchange, this favors the exchange. You can assume this and this process as generally you can see in your home. Your mother is going to if she wants to make something like clothing, so the tailor is always going to cut the pieces and he combines. So in the same way, there will be few scissors, the biological scissors like enzymes. These are going to cut the DNA and they're going to put in the other cloth. So this way a tailor is going to make a cloth beautifully. So similar process happens. This is going to occur in the biological environment, like microscopically. And next is pachytene. Pachytene stage is very important. This is a place where the crossing over is going to begin. So crossing over is, you can see the chromosomes are going to interexchange. You can see the blue color is going to match with the maroon and vice versa. So this is what called as the crossing over, and here the synapse is complete. Synapses means the joining regions, and crossing over is genetical information exchange. And again you can see the diplotene stage. The diplotene stage, the chiasmata gets visible, and then you can see erase previous things. The chiasmata will be more visible. The chiasmata are nothing but the points where exactly genetic information is getting exchanged, and then you can see it's visible here. And then diakinesis, what happens is, and then parallelly you can see the nuclear membrane is getting disintegrated slowly, and here the centromeres are ready, and the exchange is almost complete in diakinesis. By the end, you can see the recombinant chromosomes are formed. When you say recombinant chromosome, it will have the features of both. It will have partial presence of mother and partial presence of father's chromosomal mixing up. So that's what the recombinant chromosome refers to. Alright, so you can see in diakinesis the recombinant chromosomes are formed. And next you can see the next stage, the prophase 1. So this is a overview, you can say. On the other side you can see the centrosomes are being duplicated, and then the spindle fibers are being produced, and here the homologous chromosomes are quite ready. And now you can look at the metaphase 1. If you remember correctly, I told you that the chromosomes are going to in mitosis generally mitosis metaphase, generally what happens is the centromeres are going to get duplicated. I'm repeating the same step again. In case of mitosis, this centromere is going to divide. One set moves to one pole and another set moves to other pole, but it doesn't take place in this particular metaphase 1 in meiosis 1. If you see here, the centromeres are attached without duplication of the centromeres. They are being pulled away. You can see clearly, so they are being pulled away. If you look at the next step, you can understand even better. So here you can see how these are getting pulled up. There is no centromere division at all. Clear? This is how the chromosomal number is going to be reduced. So here there is only one set, so that is the reason why the chromosome number is going to come down as a being pulled up in the opposite poles. So whichever chromosome is being pulled up in the opposite poles, and they're going to form the individual chromosomes. Next step is telophase. So telophase is where the cleavage is going to happen, and the cytoplasmic constrictions are going to extend. It's going to separate the chromosomes. And here you can see clearly how the chromosomes, the recombinant chromosomes, appear after blending the characteristics of both mother and father. Okay, and the nuclear membrane is going to reappear, and we have an important term called interkinesis. The interkinesis is where the the interkinesis is where the duration or gap between one successful division to another division. So this is what important. And sometimes I learned from some of the students that even telophase 1 could be absent in order to make the gametes formation faster. And here you can see the next step is meiosis 2, and the meiosis 2 is where it is almost resembles like mitotic cell division, and it will be suffixed as meiosis 2, and then the different substages are considered or referred to as prophase 2, metaphase 2, anaphase 2, and telophase 2. Let us go through the same presentation. Okay, here we go. So after telophase we have the blended chromosomes, and it will be, remember, there won't be any cell cycle. Even if there is cell cycle, it doesn't going to go for the S phase. The S phase is completely absent. That is reason why the chromosomes are not duplicated. So this makes sure that there is no newly formed chromosome, and there is no possibility of getting 2n to 2n. Alright, so here you can see, okay, again it's in Prophase, the overview, and here the meiosis 2, and here you can see further DNA replication occurs. What I told you here, it's shown here. So Prophase 2, getting ready for Prophase 2, the chromosome shorten and start thickening, and then spindle fibers will appear, and nuclear envelope disintegrate, nucleolus disappear, and centrosomes move in opposite directions. Next is metaphase 2. In metaphase 2, those are coming in this single line of the cell plate, and next you can see the next step is where the anaphase 1, the chromosomes are pulled together. Here you can see the centromeres are getting duplicated, and those are getting moved in the opposite poles. Okay, when they get duplicated, they are moving in the opposite poles. They are almost resemble like the mitosis. Next is telophase. In telophase, there you can see the constriction, and then the nuclear envelope being reformed, and then the centrioles going back to its cytoskeletal part. That's it. In cleavage, cleavage method takes place in animals, and cell plate method takes place in the plant. Right. That's how the meiosis is going to form single set of chromosomes. That is only n. Single set of chromosomes are going to be formed from two sets of chromosome to single set. Alright, next let us look at some of the slides. So this is also one of the interesting thing you have to learn. When you say the production of the gametes in case of plants or even humans, the spermatocytes, we look at the male gamete formation here. In male gamete formation, the meiosis takes place, and all the haploids that are produced, generally one meiotic division is going to produce 4 haploid cells. So 4 haploid cells are nothing but the 4 sets of gametes, or the cells that involve in reproduction. So here you can see the male one is producing 4 haploids gametes. All of them will survive. Whereas if you look at in females, they show the diversity again. So females will produce 4 gametes, but all the 4 will not survive. So 3 of them are going to contribute the cytoplasm, and then these 3 are going to go for something called apoptosis, or they die after contributing the cytoplasm. So this way the ovule is getting bigger size. So if the ovule is bigger size, so it contributes for the growth of the fetus in a proper way. So this way, even sometimes you will be asked few questions. So how many meiotic cells will be produced in female after 100 divisions? So if it is 100 meiotic divisions, obviously only one will survive at every meiotic division. So that's why it is always equal. But whereas if it is in case of male, it will be like if there are 100 meiotic divisions in a male cell, so obviously it's going to produce 400 gamete cells. So be clear with this. So if it is a female division, it is going to produce only 100 ovules, 100 ovum, I'm sorry. And where you look at here, it will be going to produce 400 gametes. So this way the meiosis is very unique in the both male and female organisms. Clear. Let us look at the next part. I want to show you the slides. Maybe you cannot clearly see the prophase 1 substages. Maybe it may take a lot of skillful staining. Here you can see it resemblance. The nuclear envelope is getting disintegrated and it's almost absent here. And here the metaphase, they almost align in the single plate, the cell, and then the anaphase, they're being pulled away, and then you can see the meiosis 1, end of meiosis 1, those were pulled in the opposite directions. And now the second stage, meiosis 2. In meiosis 2 you can see clearly how the cells are being produced after telophase, and then gradually, I think some stages are missing here. The prophase 1, I think it's missing. Sorry, prophase 2 is missing, and meiosis metaphase 2 is here, and anaphase 2, and then the telophase 2. These were end of the single haploid sets. You can see 4 of them, single sets of chromosome. Someone missed the ploidy part. I want to tell you here, the ploidy is nothing but the number of chromosomes. Generally when you say haploid, it's nothing but half set of chromosome or single set of chromosome. It is 1n. So instead of calling 1n, we always call it as haploid. And the diploid term is seen very often. So diploid refers to 2 sets of chromosomes. So when generally these are diploid. Okay, so this is haploid and this is diploid. The reason is it has 2 sets of chromosome. One is of virginal, one is of duplicated or replicated. Okay, and then we have triploid situation as well. So triploid generally seen in it will be 3n, generally seen in case of the fruits. In fruits, the seeds covered by the food part is nothing but the triploid. Okay, so you can always see the triploid thing useful for the food. Okay, that's about the ploidy also. And then we have some nicknames of few stages of meiotic divisions. Let me show you them. I think that's it. Okay, some differentiations are there. Maybe you can even you can study from your textbook also. That's why I'm not explaining these things. Okay, let's look at the nicknames. I think prophase 1 is also called as the bouquet stage. Why we call it as bouquet stage? Because while the chromosomes are moving in the opposite directions, it appears like the, okay, I did not mention here, the prophase one will appear, the metaphase one or prophase one will be appearing like the bouquet stage. So that's it from me, Shishas. I think I want to show you even the animation. Here we go with the animation. You're ready to divide. Let you divide. I'm going to let you divide. You can see the stages here. Prophase 1 is how exactly DNA is going to combine to form recombinant chromosome. See, beautiful. Okay, next let me continue with the next stage, the condensation. Okay, the spindle fibers getting attached. Anaphase moved without centromere division. In telophase, new cells are produced. Again, the new cells are involving second division. The metaphase, anaphase. I think this is quite accurate as far as I can see. So that's it. I hope you enjoyed Meiosis learning. Please be revising it for twice or thrice. You will understand it without doubt. It's not a rocket science subject. Alright. So take care, Shishas. So see you soon with next lecture.